Okay, hello, and thank you everyone for joining us. I'm Chris Bolzan, GMGI's Chief Operating Officer, and we're so excited to bring you another installment of the GMGI Science Hour. Tonight's speaker, Pardis Sabeti, is a world-renowned infectious disease expert who has worked on the front lines of many of the deadliest outbreaks of our time, as well as on re-emerging domestic threats such as mumps. She is also a colleague and business associate of GMGI co-founder David Walt. And David tonight is going to introduce Pardis. Many of you may have attended David's talk back in mid-April when COVID-19 was yet to peak in Massachusetts. So tonight presents an opportunity for us all to get a fresh perspective from David and Pardis at a pandemic landscape that has changed dramatically in the last 2 months. Pardis will share her talk, Outbreak Response in the Genomic Age, and Davis will lead us through questions sent in advance by viewers. And tonight, for the first time, we will also be offering you a chance to submit questions via the Zoom chat function, which we will try to get to in time. If we run out of time to answer all of your questions, I'll collect them and send them to David and Pardis, and we will be sure to follow up with you via phone and email. As so many of you know, GMGI relies on the generous support of private philanthropy to hold talks like these and to help us run our operations, our research and education programs. Our great thanks goes out to all of you who have so generously been supporting us through these challenging times. So let's hear now from GMGI co-founder and board member David Walt. As most of you are well aware, David is on the faculty of Harvard Medical School in the pathology department at Brigham and Women's Hospital. And recently, David has been asked to serve on several regional and national committees that were formed to organize and guide the scientific community's response to COVID-19. And on one of these committees, he works closely with Dr. Sabeti, the Massachusetts Consortium on Pathogen Readiness. Both David and Pardis are incredibly busy, as you can well imagine, and so we are honored and grateful that they agreed to spend time with us sharing their insights tonight. So with that, I'll pass things over to David and allow him to introduce you to our amazing guest.
Good evening, everyone, colleagues and friends. I'm delighted this evening to introduce my good friend and colleague, and also one of the co-founders of Sherlock Biosciences. We heard about that a few weeks ago, Pardis Sabeti. Pardis is a professor at Harvard University, the Harvard School of Public Health, the Broad Institute of Harvard and MIT, and Howard Hughes Medical Institute investigator. Her computational genomic lab has contributed to widely varying fields, including human evolutionary biology, viral sequencing, information theory, rural disease surveillance, and education efforts in West Africa, many things that you'll hear about this evening. Pardis completed her undergraduate degree at MIT, her graduate work at Oxford University as a Rhodes Scholar, and her medical degree summa cum laude from Harvard Medical School as a Soros Fellow. She's received many awards, is well known both through her popular work and as well as her scientific work, and was named one of Time Magazine's Persons of the Year as one of the Ebola fighters back during the recent Ebola outbreak. She's also lead singer of the rock band 1,000 Days, and she's a devoted mother, and I don't know where Liam is this evening, but obviously safe and sound. So Pardis, I'm delighted to be able to introduce you, and we're looking forward to your talk.
Thank you, David, so much for being here. All over, just really excited to be here. Liam will probably make an appearance later in the call, so be looking out for that. Just warn you, you gotta let him do whatever he needs to do. So, so the least I can do as a mom. In any case, I'm really grateful to have the opportunity to speak tonight. I'm going to tell you a little bit about the work that I do, some of the work I've been doing with David, and work that I've been doing over the last just sort of many years in outbreak response. Some of it will be things that I think now has become common, part of everyone understands, but for many years trying to explain the needs. But I'll use the framing of Ebola and how we thought about it as a way of kind of laying out what are the critical needs that we have to fight pandemics in the genomic age. So I'm going to share my screen with that. Share. Okay. All right. Great. And so I'm going to just take you back to another outbreak that started in March 2014 when Ebola virus was identified as the cause of an outbreak of hemorrhagic fever, now believed to have killed nearly 60 people in southern Guinea, as government officials say. And that outbreak, which turned, was identified to be Ebola in March of 2014, is believed to have started 4 months earlier in December 2013 in a small village in rural Guinea with a little boy named Emil who was playing in a tree and inhabited by bats that got disturbed. And days later, he had a mysterious illness and passed away from it. And his sister and other family members succumbed to it as well. And then it passed through the village and into many districts throughout Guinea. But in that 4 months that passed, it had navigated throughout the country. And by April, it was already in Liberia and across the region. And then in May, it came into Sierra Leone, but it really changed. It sort of came in like a tidal wave. And if you look at the trajectory of the virus, early days, it was still one of the largest outbreaks of Ebola ever seen, was eclipsing all of the reported outbreaks before. But really in May something, there was a turn when it came into Sierra Leone. And my lab was there in Sierra Leone when this happened. We had essentially, when the outbreak was declared in March 2014, our team, 2 members of my team immediately flew out to Sierra Leone and helped them set up diagnostics for Ebola. We work at a place called the Kenema Government Hospital, which is a regional center for excellence for hemorrhagic virus disease. And we had set up diagnostics to test for this other virus, Lassa virus. We knew that if Ebola came into the country, we would see cases most likely, and that we indeed actually serve as a diagnostic center for both neighboring Guinea and Liberia. And for months we did surveillance, and then that first case emerged in May of 2014. We immediately began doing diagnostics, but then also sequenced the virus. And what we found is that in the first 14 and then 31, and in a very short period of time, hundreds of cases that were coming into the center, we started sequencing the virus and identifying hundreds of mutations that were occurring throughout the virus's genome. The viruses, they spread very quickly, and through the process, they mutate. And these mutations give us a lot of information. They inform our diagnostics, vaccines, and therapies that fundamentally are based on the genome sequence of the virus. They identify variation and identify clades of the virus that are emerging and spreading, and allow you to track the virus and see how its transmission changes. When we analyze the sequence of the virus, we could start to see what we call a phylogenetic tree, or a family tree of the virus, and we could date how long the virus had likely been in human populations. And what it appeared to be that it emerged into the human population once, likely around December of 2013, and from there was spreading from human to human. And we could begin to see distinct pressures that the virus was being faced early in the outbreak and over its course. And one of the things this kind of tells us is something about transmission. And what the data showed is that it really did enter the human population once and was spreading from human to human, not from animal vectors. And this really changed the way we thought about transmission, where early in the outbreak, if you looked at any of the messaging from the CDC or others, or around in those countries, you saw things about stay away from bats, stay away from monkeys, stay away from fruit and vegetables that have been touched by these animals. And that was really affecting a lot of people's subsistence and livelihood, when fundamentally what needed to be done, and what soon after became the mainstay, was hand washing, not touching, keeping away that physical distance, and contact tracing. It also allowed us to really start to investigate the virus. And we found that there's a very particular mutation in the glycoprotein of Ebola virus that emerged right as cases exploded and soon became the dominant lineage that spread throughout this outbreak. And as we actually began to do studies of the virus, what we found is that single mutation, an amino acid mutation on the virus's glycoprotein on the surface of the virus, was making the virus better able to infect cells, and particularly human and non-human primate cells, and made it less able to infect other mammalian cells like bats and other possible vectors of the virus. And so, and we could really begin to investigate how the infectivity of the virus changed and how it was interacting with the cell receptor of the virus. And fundamentally, the kind of implication is pretty profound. It says the virus was having more opportunities to transmit from human to human than ever before. It was improving in its ability to do so. And after that, other individuals showed this result multiple times, and it's still, and its sort of evidence is mounting that this may be the case, and also that other mutations that emerged on its backbone were further increasing its ability to infect. And that, Ebola went on to be this outbreak, and went on to be the worst case of Ebola that we have seen in our history, with over 30,000 cases and 11,000 deaths in a very dramatic way. It took the lives of many healthcare workers, which makes this more devastating. In the original image that I showed you, we were with the team of Dr. Humar Khan in Sierra Leone, and he and many of the nurses at the hospital succumbed to the disease. This really is the healthcare workers on the front lines at most risk. And so this was a tragedy, but I often talk about the fact that we were really on the precipice of a cataclysm. This is my colleague and good friend and colleague, Christian Happy. We're sitting in Redemption Church on the grounds of Redemption Camp, where Redeemers University lies. That is where Christian's is a professor, and where his hospital grounds are about 100 yards away. Just days after Dr. Khan passed away, Christian called me from his lab and told me that he had diagnosed the first case of Ebola in Lagos, Nigeria, a city of 21 million. And he ended that call by telling me that he was going to have to leave the camp as they were preparing for the main event, the first Friday, the annual first Friday of the year, where over 10 million people were going to come on the grounds of the lab where he had just diagnosed Ebola. And it was sort of a near miss within days. If this had not been detected and identified and stopped, we always think back, Christian, I think back to what if a single case of Ebola had moved through this crowd. And that really is the frame by which we have since been kind of moved to try to go as fast as we can to develop technologies to detect a virus anywhere that it emerges, to connect that information in real time across the globe, to engage individuals to come to attention, to want to seek out care and to be diagnosed and cared for, to empower every actor in the system, and to buy us time to figure out how to overcome by developing vaccines and therapies and other ways of prevention. So what does detection look like? This is the team that worked with to sequence 99 genomes from Ebola virus, working around the clock to do so. We worked in the Broad Institute and Advanced Genomics Lab, and it was a massive effort. And we could do that using genome sequencing technologies like, full disclosure, I'm a big fan of Illumina. That's just it. I'm just a big fan of it. And we use that technology broadly, but we used Illumina sequencing to generate these genomes very, very quickly in our centers. We had to do it again when Zika hit, because viruses keep coming. And as Zika spread through the Americas, we once again worked to sequence the genomes of that virus, which was a lot more difficult. And we found the same kind of patterns. The data showed us when we looked at the tree and we studied the origins, it was clear that the virus was circulating that we sequenced the virus in 11 countries. And for every country where we could date the emergence of the virus in that country, it has been in the country for many, many months, if not a year, before the first case was detected. And so there's a lag that always happens for us from when the first cases are detected and when we have the ability, when the first cases circulate and when we have the ability to detect it. So we have to really be able to do this work anywhere in the world. This is an image of a genome center that's about 90% complete. at Redeemers University. It's built by Mass Design firm, a local Boston firm that's exceptional at creating buildings, the sophisticated buildings that can work anywhere in the world, in hot environments, in low environments with issues with electricity and maintenance. It works beautifully and has a lot of systems in place to maintain that. But for the vast majority of viruses, we don't need this kind of advanced sequencing to try to find anything that's circulating, because for the vast majority of viruses, we know they exist. We've identified Zika in 1947, Ebola in 1976, SARS in 2002. We had a lot of advance notice of the possibility that this virus could circulate and the ability to have detection methods around the world. And this is actually just a PCR, a basic test that we used to detect the first case of Ebola in Sierra Leone. It's something that every lab in the region could have had as soon as the first case was detected. It's something we can scale up very, very quickly. And it is a simple method where you use assays, you design primers that target a particular sequence that you know, and can amplify that sequence and read it out using lab equipment, simple lab equipment, but we can begin to test that, and we could have that in a lot of different places. But we also need, even to go further, to really have it absolutely anywhere in the world. And Zika really showed us that, right? As Zika was circulating around the world, there's so much nervousness and such a difficulty in detecting that virus that we really need these field deployable, inexpensive, fast, rapidly programmable tests. And as David kindly served for me, showed me my conflict of interest, I want to say that I co-founded with David and others Sherlock Biosciences that builds on some of the technology I'll tell you about. But our passion is just to get these kinds of diagnostics out in the world. But we did start a company to do so, and I'm also on the board of Danaher Corporation, another company that has diagnostic testing. So take anything I say with a grain of salt. You don't have to trust anything I say here, but because I could be conflicted. But fundamentally, I just want to show you a little bit about this tech. So it sounds like you might have heard about this before, but CRISPR, I'll tell you a little bit about CRISPR for those of you who haven't. You may have heard about it because it's transforming a lot of modern medicine with this ability to use a biology of bacteria to edit genes. But really, actually, what CRISPR is and how it was discovered is it's a bacteria's immune system to viruses. It allows bacteria to detect viruses and to destroy them. And it does this by having these simple nucleic sequences, these RNA guides, that can circulate, identify a particular sequence that is innate to any particular virus. And if it recognizes it, pulls in a protein that creates a cut that does this detection and destroy. But there's a particular version of this, a particular version of the enzyme that has a neat feature. It has something called collateral effect. It once it cuts the thing that it's looking for, it starts cutting anything around it. And if you pair that with a fluorescent probe and add a quencher to it, what happens is if the cut starts to happen, it cuts the quencher, releases a fluorescence, and you can signal that something is present. If you do this in a test tube or on a strip, you could actually identify if a virus is present by looking for this signal of fluorescence. And it really can bring us to these point of care sensitive and specific diagnostics. And so these tests are, CRISPR-Cas is highly sensitive for detecting viruses and can be made very field deployable to be done on urine or on saliva and to be read out on paper. It can distinguish very specific versions of a virus as a virus changes, and it can be programmed very, very quickly. Within days of a new mutation that was important for Zika emerging, we already had a working diagnostic. And it also can be massively multiplexed. So that, we can do these paper-based readouts, but we can also put them all together in using microfluidics devices or other forms of miniaturization to do out that biochemistry in very, very miniaturized way. And so we have a chip that can run thousands of tests simultaneously. And we took 169 and then now 170 with COVID viruses known to infect humans, and we designed guides and tests for all 170 of them, and then look to see if we could identify those 170 viruses against 170 targets for those viruses. And we could see that it was beautiful. Really, if the virus you're looking for was there, it could find it and essentially would not otherwise. And it also is exquisitely good at detecting versions of a virus. If as we've heard of flu has all these different subtypes, H1N1, you know, many different versions of it, and we could actually very exquisitely identify different versions of the virus. And we tested this in the lab and on clinical samples, and it works very, very well. We've already begun to develop these tests and move them forward for COVID, and can show that both in these individual point of care tests that we are able to do this, and also to create a respiratory panel that we're now running across all of the samples in Massachusetts, in the Mass General Hospital and the Department of Health, identifying what other, if COVID is present in that sample, or if not, another respiratory virus might be present. In the context of Connect, we are able to start to think about how do we connect this information. And in a lot of outbreaks, you know, and even now here, a lot of data is shared by paper or by email. We don't have the kinds of systems, we're just sort of trying to build them and think about them now, of sharing data in real time the way we need to. So what we need is to be able to do this kind of technology at national genome centers, at regional hospitals and village clinics, but we then also need to be able to have all of that information be connected, to be shared simultaneously. And we have built systems like this in Nigeria, where we have point of care tests, hospital tests, sequencing centers, and sequencing centers across many countries in West Africa, sharing this data simultaneously. We then need to be able to engage. We need to get every individual to then want to come to attention. And some of our thinking about this came closer to home. I was on the Outbreak Surveillance Committee for Harvard University that got impaneled during Ebola. And we got impaneled again when we found out that there were infections happening on sports teams, that essentially a number of sports teams were coming down with something with parotitis, with sort of inflammation, and that were diagnosed with mumps. And then we saw that mumps was on multiple sports teams, started showing up in the dorms and roommates of these sports members, in the freshman dining hall where staff were infected, at health services where staff nurses were infected, 3 nurses were infected. Then a commencement event in which, again, students and staff were infected. And then that seeded an outbreak in East Boston. You know, Massachusetts in general has 110, Boston itself has 110 colleges, you know, 60 residential campuses. It is really can breed, you know, these residential campuses can breed outbreaks, but they can be also laboratories to think about how to respond to outbreaks. And we could actually begin to see how these outbreaks are moving between these campuses. We worked with the colleges closely, and they all agreed through a long IRB process. We worked together to say, hey, why don't we all say, this is Harvard University, this is UMass, this is BU. As long as the students can be identified, let's share what's going on and see how these infections are spreading within them. So we sequenced the samples through the schools, and we showed that there were 2 different strains of the virus that were circulating, that you know, there's this large cluster here of Harvard-affiliated infections, and we saw that this indeed what the epidemiology would suggest, or actually what the sequencing suggested and the epidemiology confirmed, was that there was this infection in East Boston that was seeded by staff that lived there at the time. And it's interesting, when we have the genomic sequence of the virus, it really is a barcode that reads out every detail of the virus, and it can give us so much information. On the left side here, you see links that are made, epidemiological links between individuals that likely say this person is somehow connected to this different person by, you know, they share something that tells that they may be, one may have infected the other. And to the right is the genomic epidemiology version of this, what the genome sequence tells you about these links. And what was great is the genome sequence really recapitulated all the traditional epi, and so what the links they thought were true, but it also then identified all these other links that you could see. And it began to make us really think about the fact that we need to be able to do this in real time, that one of the problems is the epidemiology itself is so very slow. And so, you know, and that fundamentally, it doesn't have to be that. If you think about it, you know, every time you're sick, you wonder who got you sick. Was it Janice down the hall? What did she have? You know, you start to look around. And on college campuses, it's pretty easy to think how you got sick, because you're on certain sports teams, you live in certain dorms, you go to certain parties. What if essentially you could start sharing that information together in real time, and you could start working together to figure out what your diagnosis is? And so we started working on mobile applications to share with your contacts, to share with your community what's going on, to use Bluetooth and that technology to also share that information of who you came into contact with. And this brings us to empower. We start, we work a lot in the communities. We really try to make sure that everything we develop is actually something that we can roll out and can be used by others. And so we've done a lot of programs where we've trained now over 900 individuals from the US, Europe, Africa, and Southeast Asia, mainly in Africa. But throughout those continents, we've trained individuals in diagnostics and genome sequencing, bioinformatics, project management as well, really across the piece. But we also work with students. We have a passion for, everyone in my lab is passionate about working with young students. And back in 2016, we started working with Todd Brown at the Sarasota Military Prep Academy, an amazing civics teacher, really passionate, who got us convinced to work with him to develop an outbreak simulation, a 2-week civics module on pandemic preparedness that culminates in a simulation of an outbreak. And early on, we were, it was really terrifically, they do a lot of amazing reenactments. They have big teams that work. But the one kind of tricky, clunky thing was that we were spreading the virus on paper with stickers, and that made it, it was kind of affected the realism. And as we were developing these mobile applications for contact tracing, we thought, what if we actually spread a virus via your phone via Bluetooth? What if we create a virtual virus that could create a game for the kids to try to figure out who infected whom? And that's essentially what we did. We created an epidemiological model for small populations for these test groups. And we, created this version as our first version of the app, which is a goofy version where it's sort of your smiley face. If you're healthy, then you get sicker and sicker and you can die or you can recover. And the kids can play it. And it really allowed us to learn so much about what we see, even during this outbreak. The kids were so resourceful. They ended up basically creating immunity passports. People had to show their status to get through certain areas. Then the kids started taking pictures of their healthy status so they could kind of sneak around. We saw the WHO group gave themselves vaccines before giving it to other people. The media caught wind of it. There was a fatal shooting. I mean, we saw all sorts of things going on here, but it taught us about the science as well as the cultural issues in epidemics. And what's the one kind of last interesting piece in the line of COVID was that every year, every simulation, we decide what virus are we going to circulate here, and we kind of model it after a real virus, but we try to create the drama for the students in this short event. And this fall, the virus that we were circulating both in schools as well as at multiple conferences, scientific conferences that we were running it as a module, we decided to use a SARS-like virus. And we said, but that's a little bit too easy. Why don't we make sure that it spreads asymptomatically to make it a little harder? And so we actually have a lot of data. And in real outbreaks, it's a struggle to know who is the index case? How did it actually spread? What happened here? But in these cases, we actually know, we can see exactly how it spread from the index case. So it's great modeling data as well. Found out that poster sessions at conferences are a problem because people bounce around. And of course, lunchtime is a big peak for events. In any case, and the final piece is to overcome. That essentially, for all of this, we're really trying to buy time to overcome. And genome sequencing data is so important for that too. Because fundamentally, the genome of a virus is what codes for everything that is it, that we target with our antibodies and we target with our vaccines. And so the more we know about the genome sequence, the better we can design those. For this, my own group works on therapies, and we go back to CRISPR. So we really have kind of focused in on CRISPR and the technology it can create. And essentially, as you remember, CRISPR is a bacteria's immune system to viruses. It allows it to detect viruses, but then destroy viruses. So could we use that same ability to create therapies in us? And so we started using, looking at influenza, and now sort of all of this work we're doing with COVID is to essentially say, could we design guides that could target this virus? And here we show that, in flu, it could reduce the virus itself 20-fold, and it could reduce the virus's infectivity 300-fold. So it could really knock down the virus and help individuals try to fight it off. And we really looked across the genome of the virus. We tiled the entire genome of another virus, LCMV, that's a lot like Lassa virus, a deadly hemorrhagic fever. And we wanted to see what guides, what targets would best kill the virus, and could we actually, essentially each of these guides is its own therapy. So we have all of these different drugs that we can create. We call it programmable therapies, that you can actually have one system you create, but then different versions of it you can create to target whatever you're looking for. and really began to look at lots of different viruses. And we developed a lot of machine learning approaches to study these viruses. And we could see that essentially, if you look across the hundreds of viruses that infect humans, that all of them really carry these targets, and that we could actually target many of them with many different therapies at the same time. And that's in essence the kind of work that we're doing, and all of the work that we're moving to COVID. We have developed a massively multiplex version for COVID. We've have a one-pot version of a CRISPR assay that goes all the way from saliva through to readout on a mobile application. We are using these same mobile apps to help to try to track the virus, to really detect and connect that information in real time across the grid, to engage every individual to want to come to attention. And there's a lot of technology needed for that, but obviously what the events of right now show us is a lot of trust that we need to have and to build is needed for that. And we need to empower every actor in the system so everyone feels that they have agency and understands what's going on, and to really buy time so that we can overcome. And all of this work is done with so many different amazing partners and supporters and funders. But really is fundamentally, brings me to work every day is my lab. These are images of our annual holiday cards, and I like to say that my lab commits to excellence in everything they do, and bring a lot of creativity and spark to our different cards every year. This was our last year, wishing you a roaring 20s. Unfortunately, it has been a roaring 20s, not exactly, not in ways that we would have perceived. But this is the team, and I once again want to thank you all for being here and for this opportunity to speak. Thanks.
Thank you, Pardis, for that wonderful talk. We have one live question so far, and then some ones that were submitted in advance. So the first question actually is from someone you know, Lisa Nagello-Carr.
Yeah.
She says, Pardis, what do we know about the jump into humans as it pertains to Lassa, Ebola, COVID-2, et cetera? Are there things we need to learn and know? What animal species, how close contact, et cetera? The next generation pandemics are coming. How do we predict them and identify them earlier?
Great question. It's interesting. In the case of Ebola, we don't know what the reservoir is. We believe it's bats, but sometimes, in some ways, there's more evidence that humans are the reservoir for Ebola than bats. In the sense that we see Ebola, we've seen Ebola last in humans for 500 days. In individuals that were followed, we see it replicating. We have a paper with the NIH where we show that the virus was actually replicating for over 100 days in immune privileged sites, like in the eye or in the testes, places it can hide. But it likely, if it does come in, but many things are zoonotic. The Lassa is usually coming in from rodents. The thing about it, and SARS, is believed there, to be pangolin is sort of or bats. There's multiple ways that it can come into the population. But as far as why certain ones take hold, is they may actually have features. So the fact of the matter is, we guessed to make a SARS-like virus, and people know SARS is very good at coming into humans, and coronaviruses in general spread very easily to humans. It's when they pick up a certain mutation that makes them. Coronaviruses actually infect us every year, but this particular coronavirus, there's nothing remarkable about the virus individually. It's about the combination of things. It's the fact that it, is a coronavirus that spreads rapidly, that it does have a worse fatality rate than other coronaviruses, and that it does spread asymptomatically, so it can get pretty far before you notice that it's circulating. And those are all the features together. And there is, there is some evidence and that it has picked up mutations similar to Ebola that has made it worse. There's a particular mutation in something called the spike protein that a lot of my colleague Jeremy Lubin is studying, and has been since February. And he noted that it was a virus that made that particular made it so that the particular protein is a little floppier, could allow it maybe easier entry into different tissues that could make it look like the systemic disease it's looking like. And so we're investigating these things. And that's one of the things why we always tell people you have to be mindful that like, don't ever rest on your laurels. These things are moving targets. And if it's not a big problem now, it may become one. And as far as how to detect it earlier, it really is about just having good detection in the clinics. There's a lot of people who do work around zoonotic disease, but the fact of the matter is we caught this virus very shortly after it came about. We knew about SARS-like viruses. I don't think there's much we could have done actually to be any better at discovering this virus any faster, because we already knew about it. We'd sequenced something almost identical to it. We could have had diagnostics for it. We could have had vaccines much further along to prepare for it. The genome sequence of the virus was circulated very quickly after it was, but then 4 months later, we still don't have diagnostics in Massachusetts. That is our problem, right? And there are so many different places that didn't have detections. My group just actually published data today that I didn't get a chance to get into this at the last minute, but we just published 330 genome sequence from Massachusetts that shows that we have 30 different entries into Massachusetts. This virus came in 30 different times, at least, just in the very limited data set that we have. It is circulating widely, and we are not picking it up. We haven't been picking it up in so many places.
Great. Well, related to sort of the diversity of viruses, there was a question that was submitted in advance before we get to some of the live questions, that in the past 4 months, there were something under about 300,000 deaths due to COVID-19 worldwide. How does this compare to other diseases like malaria and tuberculosis or other infectious diseases?
What's interesting about it is that, the flu causes about 50,000 deaths a year in the United States, and about 600,000 deaths annually. So far, we're still actually, so far we're actually not reaching the level that we can see from a lot of these other viruses. And it's interesting that this has caught our attention so much, and but it's kind of the constellation of things, the fact that it's new, that it's volatile, that we don't know where it's going, that we don't understand it that well. I think that's what's driving the kind of global reaction. And it is more fatal than the flu is, but actually, I said, all of those features aside, it's not orders of magnitude different in what we're seeing here. And so, in my mind, it's still a little bit of a warning sign of what even worse could come if we're not careful. But it has to be something that we stop. To me, it's the kind of frustration that you have, is that we're in this position. We didn't have to be in this position. We didn't have to social distance if we had just been more prepared. And to this level, obviously, to this extent. And so, I think these are the kinds of things where I always say, like, we should have. I'm always the one in the office saying, like, people need to stay home when they're sick. I was trying to get we've been terrible about social distancing in general. People come in fully sick. This is going to change a lot of that paradigm where we won't want people coming in sick. And those kinds of things we can change and get much, much better about. And then to your point though, to your question, David, the fact of the matter is malaria kills about 1 million people a year. These other things are infectious diseases are the biggest, among the biggest killers out there of human populations. But what is amazing about them is that they all kind of operate the same way, and the same technologies that we need to think about malaria many of them are applicable to viruses and other things, and definitely across viruses. All of these technologies we're building for COVID could help us in fighting the next seasonal flu, fighting Ebola if it comes again. And so it's really a shame. Why were we not invested beginning? We could have actually helped seasonal flu every year that causes not only mortality, but incredible amount of morbidity and lost economic, a lot of economic loss. We could have done this before. We didn't need to get to trillions of dollars of debt and a global crisis to get to the point to know that we should invest more.
So I actually like to add to that a little bit, because both, I think, malaria and tuberculosis in particular, probably there will be more deaths this year because of COVID, just because of lack of access to antibiotics, lack of access to healthcare. So I think that those millions of deaths per year from those diseases, and with TB, it's mostly children. Unfortunately, it's I think begun COVID, I think, has brought to the first world the issues that the developing world faces every year. And I think that's, something that we really have to think about carefully in terms of how to not just help ourselves, but to help others as well. And COVID now is going rampant through Africa and other parts of the world. So a few questions coming in. One that's near and dear to our hearts. When will Sherlock release a quick, easy, cheap detector for SARS-CoV-2?
David, we can do this Q&A together. Do you want to take that one, or?
No, you could do it.
They're working on it. The Sherlock Biosciences has an FDA approved diagnostic already out there. It's the first CRISPR-based diagnostic to be FDA approved. And there's a lot of technology development in the company and around the company, that is moving that forward to, we ourselves have this, we've just published it on the bioRxiv, a one-pot assay, essentially something that can go from saliva out to paper or fluorescent readout, that can happen within a short period of time. And it's a lot of it is that it's still a nascent technology, although it's really going through hyperspeed right now, where it's like a totally new form of a technology, but we're moving it very, very quickly to try to get it to that kind of manufacturer scale. But what's powerful about that technology is it's what I call a platform technology. There are a lot of detection methods that are really good at being, can be rolled out in the field, like a pregnancy test type of thing. The issue with those is that when they're based on proteins, they're pretty bespoke and each one has to be developed over time. With PCR or with CRISPR, it's literally just, you type out the actual nucleotide sequence you want to target and synthesize that. And you immediately have a diagnostic. You can have a new diagnostic in a day. And so as we're taking a little time to develop the ability to have our first rapid point of care test, once you have that in hand, you can have it for anything very, very quickly. And that's going to be really exciting.
Okay, big picture question. What's the most important thing we've learned from the current COVID-19 crisis that will help to prevent the global spread of the next emerging pathogen?
I mean, we've learned a lot, and again, what's funny is we both learned a lot, and then also all the people who've been in Ebola and Zika before are like, but we always knew this, we're just doing it now. We learned that contact tracing matters. It has to be done very, very quickly or sort of pointless. We've learned that diagnostics are critical, and that's actually a big boon for folks like David and I, because it's really it's very hard to convince people that diagnostics matter when all of the funding in pharma and everything else goes to therapies. But it's like, if we just detect it early, we won't need these expensive therapies, particularly in infectious disease. If you can prevent it, you can really save a lot of life lost and cost on the other side. So I think just understanding that, just understanding this sort of contacts and engaging with the community, with the fact that diagnostic tests matter, the fact that we can actually work as a community, and we're seeing some success stories of working as communities, and we're seeing also the trials and tribulations of being in an outbreak. I wrote a book with a journalist, Laura Salahi, called Outbreak Culture, and it's really about the toxic culture that emerges during outbreaks when an insidious, deadly threat is moving between individuals, and you start getting fear and paranoia and human instincts for survival, sometimes at a cost to other people. The things that we saw in our game with the kids, and we're seeing actually the tension that rises, the suspicion, the paranoia, and then bad actors who are truly making profit out of the system. That it's a struggle. There's a lot of things that can go wrong, and we have to. I say, and I've just wrote an op-ed that'll be coming out in Stat Opinion soon that sort of says outbreaks truly exploit the cracks between us, the cracks in the trust between us, and they amplify them. And that's what we're seeing here, we already had things bad in the way in our issues with race in this country. They got even worse in sort of the tense environment of an outbreak. And now the virus is actually exploiting the response to that even further. It's just a mess. We have to get it together between ourselves. This is one place where it's a war, but it's a war of individuals together against a common threat. It's the one place we should figure out how to work together. But it just doesn't always happen that way. You have to commit to that.
We saw this with the competition for Purell.
Yep, yep, yeah, definitely, yeah,
Um,
yeah, punching each other for supplies. I mean, it's
yeah,
yeah.
exactly. Um, Speaking of therapies, a question about, do you have a COVID therapy in the works? I think you alluded to that. Is that being contemplated as a therapy?
It is, yeah, and we and others are working on those, developing them. There's still a ways to go for those. They're a little bit further away in the sense that they still have to. That's another great platform technology. Once we figure out how to get it safely into humans and working well, then we're in a place where it could be, you know, a new therapy could be created within a day in that same way. But there's still some here to there to figure out how to safely get it into humans, into the right tissues. But we're working on that for sure.
So something that's non-COVID, could you comment on the use of genome sequencing related to the potential development of a new vaccine for mumps? As seen during multiple outbreaks in college and other communities, 2 MMR vaccines were not as protective as we would like. The targeted use of a third MMR is recommended during mumps outbreaks, but efficacy is not convincing. Thank you.
Okay, yeah, all right. Yeah, so we actually investigated a lot I have two papers. One this of the genome sequencing, where we really looked at it, and we also, studied the target of the vaccine and said, does this look like vaccine escape or anything like that? And it doesn't. It just looks like waning immunity. It does look that the vaccine is not something you don't have to do annually, but you have to do a couple times in life, and a third MMR would have been recommended. The fact of the matter is, though, the vaccine is still helpful. It may not protect you from getting mumps, but it definitely makes it far less serious in the sense that prior to the vaccine, there was infertility and other kind of very serious complications could happen from mumps. Where here, that's not what we were seeing, and so we studied and saw exactly, saw that this was really about waning immunity, and the idea that a third vaccine would be useful. And then in another study, we really started investigating the the dynamics in Harvard University, and then kind of did a trade-off of what would be the cost of doing. It would have cost $80 million to revaccinate the whole population, but $75,000. We had a very small thing of how we could have stopped it from. Mumps doesn't spread as easily as COVID. There's very cheap things you can do to do detection and prevention, and so we sort of made the case in that detection and prevention is the better way to go.
Okay, thank you. I believe SARS-CoV-2 has a proofreading mechanism, whereas influenza doesn't, leading to lesser or greater mutation rates. Where does Ebola fit in that spectrum, and how do you cope with the greater mutation rate of some viruses?
What was it? It was SARS, Ebola, and what was the other one that you mentioned?
There was nothing else mentioned. It was just that influenza.
Oh, influenza. Yeah. Yeah. Influenza has, I should know, I usually know the substitution rates of all of them, but SARS, right, so far, we were just looking at the complete set, and there are only 41 mutations that are greater than 1%. There are mutations, they are emerging. These are all RNA viruses, and they do, and lots of mutations pop up, but most of them kind of disappear. But we're not, there's not a massive amount of diversity. Something like Lassa on the other side, 1 in 3 bases, 1 in 3 nucleotides is different. I mean, those viruses are just tremendously divergent, which make them very complicated for diagnostics and therapies and beyond. But Ebola and SARS seem to be a little bit less variable in that way. But that still doesn't mean that a mutation can't occur and that a mutation can't be impactful. And it doesn't mean that a mutation can't disrupt a diagnostic target. So all of that, means that it's still important to sort of track what's circulating.
Okay, a question from Barb Ennis. As a lean, all caps, practitioner, it is interesting to understand how important tracing is. It makes sense. It is key to your studies. Why are we in the US just hearing about tracing?
Why are we? Because no one's been listening to me talk about it for 60 years. I guess you're not on my on my circuit. Yeah, I don't know. What's interesting is that we haven't seen a lot of people talk about it before in general that much, and a lot of these folks are building things in real time now. I mean, contact tracing in general has been a very classic thing. I think the use of Bluetooth technology and more rapid ways of doing it is more recent. And actually, I should also disclose that I filed patents pre-COVID, to use all of that technology. Basically, I filed patents, but both for using Bluetooth and contact tracing information to rapidly diagnose infectious diseases. So that's another disclosure. But in that case, I very specifically, once we started doing the game with the students and we saw how sideways it could go, it's not something I'd ever really file a patent for. We actually filed a protective patent because we thought it could go sideways and we didn't want companies like Facebook and others to have it and use it in bad ways. And so part of the op-ed that we're publishing tomorrow is about that. It's about the fact that if you're going to create them, they have to protect the most vulnerable. And we don't plan on we don't plan on if we get this patent, and we filed it a year ago. We don't plan on enforcing it unless somebody uses it for ill will. And I think it is really, really important that they do so, because we are seeing the many ways that it is being misused. Even in a recent event in Seoul where the individual happened to go to LGBT clubs, and then suddenly people are being outed for their sexual orientation, and we see them being used in protests. And so again, this is only going to work if people trust the system and trust how it's going to be used.
Okay, two more questions. The one was a pre-submitted question, and it actually relates, I think, to the sort of the mission of GMGI, and that we are using these new genomic technologies, in particular sequencing, to uncover information that was not available previously by mining data from the ocean. From your perspective, how have advances in scientific technology between 2014 and today affected our responses to viral outbreaks, for example, between the Ebola 2014 outbreak and COVID-19?
Yeah, I mean, I think that we're much faster at sequencing, much faster at generating data, and sharing, and even thinking about it, like even a lot of the tech, like Zoom is better than it was before, and Slack and text and all these things have advanced. They really put us in a much better position than we would have been. Or the fact that bioRxiv has moved so fast allows us to share our research results very, very quickly. And so we are very fortunate in many ways to have had this event happen in a time when we can communicate and cooperate and coordinate. So we're better positioned than we ever were. It's just still phenomenal that we've known about SARS since 2002, and we could have had all of this stuff in place, and we knew everybody's known it's a threat. I mean, that's the kind of funny thing. It's like, so we decided to make a SARS-like virus that spreads asymptomatically. Looks like we are, and we actually did that exercise in China, in schools in China, as well as in conferences. It looks pretty bad. It looks as if we were like, conspiracy theorists that might say that we knew what we were developing, or we were testing something out, but it's just not true. I think that, the movie Contagion essentially used a SARS-like virus too. I mean, everybody knew we should be looking out for SARS, but yet here we are. And everybody saw this thing emerge in Wuhan, and yet here we are. And so we're in a place where we could, move much faster than we ever could before. But at the same time, we also lost so much time unnecessarily.
Okay, last question comes from me.
Okay.
Can you describe for the listeners what's going on in the Boston area with respect to COVID-19 and the scientific community?
Yeah. Oh, I mean, what, in the good ways or in the bad ways, or in the...
What's been going on?
Well, I mean, there are, COVID is spreading in Boston, and it's one of the worst hit places. It's one of the top, 5 worst.
I was asking about what the scientific community is doing.
Yeah. No, I was going to start it there. Yeah. Don't worry. I was going to end on a high note. I was going to start with a low. It's bad. There's bad things happening. But the scientific community here has come in with a gusto, one of the most, it's this weird thing in outbreaks, because in one side, it's like terrible and horrible things are happening. On the other side, you make these like fast friends that are now your best friends. David and I, met each other a few times before COVID. He's now like on my like, we're texting it all the time. If I don't see his face, many times in the morning, at night, I don't know which way is up. So it's an amazing thing. We're just immediately you build fast friends and bonds with people who are terrific. And we are co-chairing the Massachusetts Consortium for Pathogen Readiness, Mass CPR Diagnostic Working Group. And it's been one of the highlights of my career to do this, to kind of get together and together try to navigate all of this landscape. We started our working group, and we're like, okay, let's think about all these technologies and research. And our constituents basically said, like, I don't want to talk about research. I don't have diagnostics. So we had to first stop and say, how do we make sure all these places have diagnostics? Let's stop the stopgap. Let's make sure that we stop the bleeding. And then from there, okay, now everybody's kind of got a baseline to work from. Now let's bring all this cool tech together and see what we can move. And then now we're bringing in all sorts of other partners around implementation and thinking about how to get businesses back. And so it's been a lot of fun to see all of these colleagues that I've been in this community with for the 12 years that I've been a faculty and the 26 years since I've been kicking around this area. But now suddenly the whole band's together and people are working together in a really tremendous way. And so there is a lot of positivity that can be seen from this, and a lot of negativity. You really do see the best and the worst of of humanity during an outbreak, and we have to just keep committing and pushing ourselves to our better selves.
Well, Pardis, I have to thank you immensely for joining us this evening and for really sharing your experience and just being able to convey all the incredible information that you did tonight. I'm going to turn it back to Chris to wrap it up, but really on behalf of the community, we really appreciate your taking the time from your busy schedule to share your knowledge with us and your experience.
Well, my pleasure. And thank you, David. Thank you, Chris. And thank you, everybody who took the time on a Thursday evening after a long week.
I think Chris is going to take
Sure.
up. Oh.
Yeah. Thank you both so much. I echo David's comments, Pardis. It was incredibly helpful, insightful, exciting. And David, we appreciate all of your time as well. We know you're both incredibly busy, working so hard on behalf of all of us. And it's such an incredible honor to get a peek into the work that all of you are doing. So thank you both so much. We did have one last question. Pardis, folks are wondering when your band is playing next and where do they play?
Yeah.
When those things open up again, GMGI will be sharing.
Yeah, God, bringing back live music, that's going to be an interesting one. So yeah, don't you'll be a little ways away. But
Okay, well, we look forward to that. We look forward to seeing all of you in person when we can do those things once again. We do have a great slate of speakers lined up for the whole month of June. So Ashley will be sending out to all of you that lineup and an invitation on Monday. So get ready. We've got some other really great, exciting talks coming up really soon. We've recorded tonight's talk and we'll make that available to you starting tomorrow. So if anyone else had any other questions, I did manage to record any ones that we weren't able to get to. So thank you all so much for tuning in. Thank you again to David and Pardis for spending their time with us on a Thursday night. We're really, really grateful. This was so amazing. Email us with any questions that you might have that didn't get answered or any other thoughts or feedback. Like we just said, we all really look forward to doing this again in person at 417 Main, where we like to gather for our science talks. But until then, please everybody keep well and have a good night.